name is Anny Cardenas and I work as a postdoctoral fellow in Professor Christian's Wolster lab. I am a marine microbiologist and my particular interest is to understand bacterial communities that are associated with corals and specifically to understand their beneficial impact on the coral health.
Korallenriffe sind faszinierende und einzigartige Ökosysteme und Lebensräume für eine Vielzahl von Pflanzen und Tieren. Durch den Klimawandel und den dadurch hervorgerufenen steigenden Wassertemperaturen sind Korallenriffe aber besonders gefährdet. Korallen leben in Symbiose mit bestimmten Algenarten, die insbesondere für ihre leuchtenden Farben verantwortlich sind. Diese lebenswichtige und fragile Symbiose kann bei steigenden Wassertemperaturen zusammenbrechen. Frau Cardenas erforscht mit ihrer Arbeit den Zusammenhang zwischen Korallengesundheit und dem Vorhandensein von bakteriellen Gemeinschaften, die in räumlicher Nähe zu Symbiodiniaceae-Zellen leben, denn diese sind für die Umweltanpassung der Algen von grundlegender Bedeutung.
Corals are made up of hundreds to thousands of individual polyps that secrete their own skeletons of calcium carbonate to form the structures that we know as coral reefs. And one of the most fascinating aspects of coral biology is the symbiotic relationship they form with photosynthetic microscopic algae, which are responsible for providing most of the coral's nutrition. Bacteria that live in coral tissues can also provide beneficial functions to the coral and to the algae by recycling nutrients, providing vitamins, defending against pathogens to improve coral's health, and to mitigate environmental stress. But very little is currently known about the beneficial groups of bacteria and the mechanisms they use to support coral health. Corals are one of the most valuable ecosystems in terms of biodiversity and services they provide to us. Coral reefs are an essential habitat for more than 30% of the world's named marine species, and they provide food, livelihoods, coastal protection, and tourism to over 500 million people living in coastal areas, making them a valuable economic source. These ecosystems are unfortunately threatened by climate change, and one of the primary causes of coral reef decline is coral bleaching caused by global warming. Temperature increases in seawater can cause the coral-algal symbiosis to break down, and without their algal symbionts, coral would not only lose their color but also their main source of nutrition. So most coral bleach at 1 to 2 degrees above the average temperatures of the hottest month in that specific location, but prolonged heat waves can cause severe bleaching events that result in mass coral reef death. The decline of coral reefs has a catastrophic ecological and economic consequences for us too. Therefore, we need to better understand the mechanisms of coral thermotolerance to be able to assist in the design of innovative approaches that can help tackling the coral reef crisis. Microscopic algae play an important role in their coral host thermotolerance because some of the algal species are more tolerant to high temperatures and allow their coral hosts to survive in places with hotter seawater temperatures. So we want to look into the bacterial communities that are closely associated with these thermotolerant microscopic algae, and we want to see if bacterial-algal interactions have an effect on the algal physiology, which could have an impact on the algal's ability to mitigate thermal stress. We recently participated in a research trip to Saudi Arabia to the King Abdullah University of Science and Technology, where we sampled different species of corals that presumably harbor different algal types with different thermotolerance, and performed heat stress experiments to diagnose their thermal tolerance and identify the most heat-resilient coral colonies. We will be conducting cell fractionations that allow us to isolate the algal cells from the coral cells, and then we will characterize the bacterial communities associated with the algal symbiont, as well as the genomic features of these bacteria, which can provide us with clues about the beneficial mechanisms by which they can improve algal thermal tolerance. Thank you very much to the Werner and Erika Messmer Foundation for funding this project and for promoting early career researchers like me.
Die Messmer-Stiftung stellt Wissenschaft und Forschung in den Vordergrund und wir sind der Überzeugung, dass sie mit ihrer Arbeit einen ganz wichtigen Beitrag dazu leisten kann, zum Erhalt dieser beeindruckenden Lebenswelt beitragen zu können. Liebe Frau Cardenas, herzlichen Glückwunsch zum Wissenschaftspreis der Werner und Erika Messmer-Stiftung.